Good morning, everyone. Welcome to Beth Israel's event on Israeli innovation and responses to COVID-19 as part of the function of Beth Israel's Israel Advocacy Committee. I am Ruth Sherman, one of the co-chairs of the Israel Advocacy Committee. Other co-chairs are Martha Barvin and Jeff and Shelley Lewis. Today we're going to start off with Nadav Kidron. Nadav, thank you so much for being with us.
Thank you for having me, and nice to chat, Ruth.
Oh, I'm looking forward to it. By way of introduction, Nadav Kidron is Chief Executive Officer, President, and Director of OraMed Pharmaceuticals, founded in 2006, and its subsidiary, OraVax, founded in 2021. Mr. Kidron is an entrepreneur whose experience includes senior executive roles in a wide range of industries. He holds a bachelor's degree in law and an international master's in business administration, both from Bar-Ilan University in Israel. He is a member of the Mirage Business Executive Leadership Program and a member of the Israeli Bar Association. He is an international lecturer on Israel's entrepreneurial culture and the country's roots as an oasis of innovation ideas. Again, Nadav, welcome. You are developing an oral COVID-19 vaccine. What inspired you to begin on this adventure? Why do we need a new vaccine?
Okay, so COVID-19 is a very, very trendy thing, and we all are busy thinking and wanting to know what's going on with COVID-19. I cannot think of anything else that affected our lives as much as this COVID-19 story. We all wanted to make plans. We're thinking of making plans, people who have young kids. And I'm a firm believer that the solution to many of our challenges has to be derived from a new innovation and technologies. And the same goes here. So you may ask me, wait, but we have the Pfizer and Moderna's vaccine. But then if you look around us, okay, and we're almost at the last day of 2021, I'm now in the United States. I cannot go to Israel because it's a red country. In Israel, they're about to get their fourth booster vaccine. You know, my kid's school is probably closing down and they're going to be moving into Zoom. So obviously there is still something that's lacking, meaning we have a solution, but it's not an holistic solution. And that's what brought us to develop something that can be better than what we currently have.
Oh, excellent. Very good. What are the advantages of an oral vaccine? What is it that makes an oral vaccine an absolute game changer for the globe?
So maybe, Ruth, I'll take this opportunity to share with you a short video that will show exactly why is it that, you know, we are developing that vaccine, oral vaccine that we're developing.
Imagine a world with an oral COVID-19 vaccine. As the world races to vaccinate people against the COVID-19 virus, the effort is hampered by the need to keep vaccines at freezing temperatures and have qualified people administer the shots. Imagine if the shots would be required every year or every 6 months. But what if the vaccine could be taken as a pill in the comfort of your own home? An oral vaccine could help quickly vaccinate the most vulnerable and people in remote areas. OraVax, a company based on OraMed Pharmaceuticals' patented oral delivery technology, is developing an oral COVID-19 vaccine. OraVax's vaccine in development, should it be approved, would not have to be kept at below freezing temperatures and would not have to be administered at central locations. An oral vaccine that could pave the way to a pandemic-free world.
Okay, so I think that gives a sort of a little bit of an overview about the oral vaccine, but let me also state the obvious and the non-obvious. So the obvious and the reason that an oral vaccine makes so much sense is because there are many people that will take a vaccine, if they will take it orally, versus an injection. Just in the United States, a survey was made, and about 19 million Americans would take an oral vaccine and are not taking the current vaccine that they need to inject. But the advantage goes ways beyond that, because an oral vaccine means that you don't need a professional to administer it. So it's much easier, logistically wise, to give out the vaccine. An oral vaccine also means that it doesn't have to be at a freezer temperature. So it's much easier logistically to take it to one place to the other. An oral vaccine means that, unlike the injection, you don't have all the waste, the billions of syringes and plastic and all of that. You swallow a pill and you're done. And an oral vaccine means less side effects. Many people, especially on the second and third booster, they feeling almost like they getting the corona after getting the vaccine. But if we give something through the oral route, we're not going to have those issues. But last, and probably the most important thing about an oral vaccine, and this is the most critical point when I said that we need to look into innovation in order to be able to deal even better with the current crisis, is that the oral vaccine that OraVax has is made out of 3 proteins. And therefore, it should be able to give us that immunity and protection, not only from the existing variants, but also from future variants. And the idea is that you take the vaccines, you vaccinate it, the same thing that when we're 6 months old and 1 year old, we got a vaccine, and then we're done. The T cell will remember it for the rest of our lives, and life can go back to the way it was 2 years ago.
That's so very exciting, Nadav, just to even see the film. And I hear you saying that that your vaccine, covers the possibility of more mutants, and also the idea for underdeveloped countries, what a difference it makes not to have to worry about the refrigeration aspect is like really a game changer. It's very, very exciting. Tell me, the 2 predominant vaccines present now are messenger RNA vaccines. Tell us, is your vaccine a messenger RNA, or tell us a little more about that?
No, so we're using a VLP technology. So basically we're going like the old good days. And the innovation is that we're able to take the S spike, the E and the M, and combine them together. And then what we do is we give it, and obviously when we give it orally, but potentially we could be giving it as an injection as well. So you actually have a much, much stronger protection, because Pfizer and Moderna with the messenger RNA, they are using only the S spike. And by us putting together the E and the M, you have to remember that the S spike is the one that most easily get mutated. But that's not the case for the E and the M. So it's like we're having 3 shields that even if there are going to be mutations to the S, we're still going to provide the protection from feeling any sickness, because of the idea that we put the E and the M, and we're going to have the antibodies for the E and the M in the people who actually took that vaccine. So that's why it's as classic, it's safe, easier and cheaper to produce. And that's why we believe that that's the direction we should be heading.
Ah, wonderful. So it covers more virus parts like the protein envelope and the membrane as well as the spikes. So you have a much better chance of covering any mutations that come up. It's so exciting. That's just wonderful. Now, the big question, Nadav, where did this idea come from? What's the background of the development of this very unusual vaccine in the history of vaccines?
So our story starts many years ago in the diabetes department at Hadassah Medical Center. When I grew up as a child, I knew that my mother, she's a researcher at Hadassah Medical Center working on alternative ways to deliver insulin. About, I'd say, actually now it's almost 16 years ago. 16 years ago, after many years of research, my mother told me, listen, son, finally we had a breakthrough and we can deliver the oral insulin, or the insulin, we can deliver it orally. So I basically, my background is I'm a lawyer and an MBA. I basically stopped everything I did, took the intellectual property out of Hadassah Medical Center and established OraMed, where OraMed is a company that have a platform technology that allows us to take injections and deliver them orally. And our flagship product is insulin. But we have this strong platform technology that has been proven undergoing a phase 3 with the insulin in existence. Then, in another side of the world, in India, there's another company called Primus. Primus is one of the leading biotechnology developers out of India. And they came up with this VLP vaccine that can work against the variants. So we put the two together, and a newborn came to the world called OraVax, for oral vaccine, which took these two technologies, put them together, and now we have a 3-protein vaccine that can be administered orally.
That's just wonderful. That's an amazing story of wonderful collaboration between Hadassah Hospital, your mother, and the company in India producing virus-like particle, a VLP, and we have something very, very, very unusual. This is a terrific story. It really, really is. Can you tell us some more about your work as far as changing diabetes? We're talking about insulin that does not have to be injected any longer by this product. Tell us more about that and your role in that development.
So maybe if you don't mind, I will share another short video to show exactly that.
Imagine a world
That's
with
not
an
that one, right one. Let me just get the right one. So I want to share a small short video that will show us what is it that OraMed does.
Diabetes is a pandemic with over 500 million patients. It is the single most common disease that the human race has ever had to face. From the first day insulin was injected, people try to figure out how to put insulin in a pill. OraMed's technology allows us to overcome those technical obstacles and to deliver those injections, those peptides orally. So we're talking about a safer insulin where patients take a pill. What we are doing is something that can better the lives of hundreds of millions of people around the world.
So the idea is really, when we give insulin orally, it's not only, it's easier, patient's compliance is higher when you take something oral versus an injection. The idea is that when we give it orally, it has a first pass into the liver. And the liver is the organ that regulates the secretion of the insulin, of the glucose, but we're able to mimic the physiological way that the body works.
I see. So by physiological way, through the digestive tract into the bloodstream, around then to where insulin normally is. So that is just fantastic. Anybody who's had a family member that you have to watch several times a day, measuring and injecting themselves. And as you say, disposable syringes, the whole thing. It's just amazing. That's a really wonderful story. And, this is fantastic. I wish, you continued success in all of this. It's just amazing and very unique, really innovative. I have one last question that I would like to ask you, because I know that you have been very interested in explaining to the world about the Israeli innovation culture, and that's what this seminar is about as well, Nadav. So we'd love to hear your view of the Israeli innovation culture and how this impacts new innovations in healthcare.
So, many times as people who believe, we look back at the time of the Bible and different things, and we say, how did people they saw these miracles, how come they didn't recognize them? I think when we're seeing the innovation that comes out of Israel, we're seeing a miracle. Only this year, we're about to end the year 21. almost $25 billion. $25 billion were invested in Israel innovation. We're talking about a small country that has an impact way larger than what you would expect from such a small country. And the good news is that it's growing every year, beats the previous year, and it's continuing to grow, and people getting better in it, and getting better at doing those international businesses, and getting better on building these larger companies and more unicorns. So I really think that we're witnessing a miracle, and I invite everybody, when you come to visit Israel, don't only go to the Western world, don't only go to Caesarea, don't only go to the fine restaurants, but try to take some of your time to be exposed to some of the most amazing innovations that are coming out of Israel and are basically impacting everybody around the world, anybody who's sitting around the table right now.
That's just really amazing, such a small country, so much innovation, and it's noted the cooperation. You're working with a company from India, you know, everybody's collaborating with everybody else. There's not so much keeping your product a secret and so forth and so on, but everybody in Israel wants to help everybody else. And traveling around, they may be talking about their company, but they're going to talk about other things too, about Israel innovation and really promoting the country. It's a totally beautiful story, Nadav. It's just a pleasure to have had you here today to talk about this very, very exciting work. I can assure you that the people who are watching are going to be watching your progress and, you know, hoping the very best for you for continued rapid growth, as this is such a necessary product. And thank you so much for being with us today.
Thank you, Ruth, and good luck there.
Thank you, my pleasure. Good morning, everyone. I'm Ruth Sherman, one of the co-chairs of the Israel Advocacy Committee of Congregation Beth Israel. And our topic of the webinar this morning is Israeli accomplishments in the struggle with COVID-19. We're very honored to have with us Professor Eran Segal. And welcome, Professor. Glad to have you with us. I'm going to take a few minutes to introduce Professor Segal to the participants. We are here today with Eran Segal, who's a professor at the Department of Computer Science and Applied Mathematics at the world-renowned Weizmann Institute of Science in Rehovot, Israel. Professor Segal leads a multidisciplinary team of computational biologists and experimental scientists. His research focuses on nutrition, genetics, microbial populations within the body, and the effect of these factors on health and disease. His aim is to develop personalized medicine based on the accumulation of data from large samples of human subjects. Professor Segal was awarded a BS degree in computer science, summa cum laude, in 1998 from Tel Aviv University, and a Ph.D. in computer science and genetics in 2004 from Stanford University. Before joining the Weizmann Institute, he held an independent research position at Rockefeller University in New York. Professor Segal has published over 170 publications and received honors and awards for his work, including the Overton Prize for Outstanding Contributions in Computational Biology. He was elected to membership of the European Molecular Biology Organization and also as a member of the Young Israeli Academy of Sciences. Relative to our seminar today, that of COVID-19, during the course of the pandemic, Professor Segal developed models for analyzing the dynamics of the pandemic and served as an advisor to the government of Israel. Again, welcome, Eran. And
thank you.
yes, serving as an advisor to the Israeli government on COVID-19 pandemic. How would you describe the struggle of Israel with COVID-19 throughout the four waves of the pandemic?
Yeah, wow, that's a very big question. It's been it's been a lot that I guess we and many other countries, all the countries in the world have gone through. I would say I would divide it into maybe two main parts. One is before the vaccines and then after the vaccines. I would say before the vaccines, during the first wave, nobody knew what was going on. So I think Israel rightfully decided to shut down its borders as soon as the pandemic hit. And so in retrospect, we had a very, very small wave. If you look, if you put all the waves of Israel in perspective now, you'll barely see that there was anything during that first wave. And so we got out of that wave. We were in lockdown during the holidays. This was in March of 2020 during Passover. And then people thought, like in other places in the world, that it's behind us. And our Prime Minister, Benjamin Netanyahu at the time, even told people in May of 2020 to go out and celebrate. But of course, then Israel was hit by a second wave. There were no vaccines at the time, obviously. This is in July of 2020. At that time, the interest here shifted more towards trying to make sure that we don't reach a situation in the hospitals where we're overloaded and we don't have capacity in the healthcare system. And so the magic number was 800 critically ill patients at the same time in the hospitals as kind of being the bound that was declared by the medical doctors here, that if we pass that, treatment is going to be reduced in its efficacy. So being a computer scientist by training, myself and my lab, we joined the efforts and tried to develop models to predict what would be in the future, and say 1 or 2 weeks ahead of time each time, what would be the number of critically ill patients that we'll see in the hospitals. So the models we developed were quite accurate. We tested them in real time. I used to post on my Twitter account ahead of time. These are our predictions. I gave them also to our government, and they were quite accurate. And then before the holidays, which were in September of 2020, we saw in our models that although we were at around 550 critically ill patients, we're actually going to pass the 800 number, because as you know, you always have a 2-week lag at least between any action that you take until you see its effect. So we said, even if you do a lockdown today, we're going to go over that number. And when I showed that to the government, I guess that together with other considerations led Israel, again, Benjamin Netanyahu at the time, to declare a second lockdown to combat the rise. So during the holidays, the high holiday season, we were in lockdown. That managed to lower the numbers. And then we gradually got out of lockdown, still not with very low numbers, but we got out of lockdown. And then what hit Israel is its third wave, which came about with the Alpha variant, the variant that emerged originally probably from the UK. And I guess we did not do a good enough job at shutting down the borders, and the Alpha variant came and it started to rise. And then there was a really big dilemma, because it started to rise at about the same time when vaccines were beginning to be administered also here in Israel. But when we did the computations at the time, and according to our models, we saw that because the Alpha variant is much more transmissible than the original variant from Wuhan, then according to our models, we were going to go way above the 800 and actually even above the 1,000 hospitalized patients, which was considered to be really a place you don't want to go to. So we conveyed that also to the government, and then Israel declared its third lockdown, which I guess even in retrospect was a good decision to do at the time, because we still reached 1,200 hospitalized patients. We published papers and did analyses to show that, unfortunately, even when we pass 500 or 600 critically ill patients together, the quality of care goes down in the hospital. So more people probably died than they should have if we did not have such a high load on the hospitals. So the decision of lockdown was the right one, even though we started vaccinating, because the rate of vaccine was not enough to overcome the exponential growth of the Alpha variant. So what happened then is that the wave began to go down, and I would say until this point, this is kind of the era before vaccines, and I think Israel did probably I would rank it a mediocre job. It wasn't the worst of all countries. There's countries that dealt with the pandemic worse, and it was certainly not among the best. Which at that point, before the vaccines, in retrospect, my view is that countries like Singapore, like Taiwan, like Australia, New Zealand managed the pandemic much better in the era before vaccines, because they paid a higher price when the rise just began, because they took strict measures, but they benefited then from having long periods of essentially almost no COVID, where they could have a really open and normal life without much restrictions. But I would say from that point on, from the era of vaccines, which came about in January, I think Israel since then has done an extraordinary job and has actually been teaching the world a lot about the effect of vaccines. And it really became a model for how vaccines should be rolled out. I think we still could have done a better job, but we did, I think, a superior job, a really good job, because of the centralized healthcare system that we have here. There's only 4 HMOs in Israel. By law, each citizen has to be a member of one of these HMOs, and there's a central central database by the health organization. I don't think that exists in the United States, for instance, or in many other countries. And that really allowed a very rapid rollout of the vaccination program. At the peak, we vaccinated 250,000 people every day, which in the US would be, I guess, the equivalent of maybe close to 8 or 9 million people per day. So it was really very, very rapid. And that brought down the third wave. And Israel then opened up around February. It started to open back and lift restrictions, which gradually were lifted until in May, all of the restrictions, including at the very end, I think around June, also the requirement for masks was lifted. And Israel, if you look at the graphs, was ahead by maybe 3 or 4 months in terms of hitting the rate of people being vaccinated compared to the rest of the world. So that was I think, very big achievement, and it allowed life to come back to normal quicker. And then finally, in the fourth wave, everybody was quite surprised, because we really thought we were over it. We knew that new variants could come, but we knew that we're also vaccinated and we may have reached herd immunity. But then something that surprised all of us and surprised the world, and again here Israel led the way in terms of the discovery, is the discovery that these vaccines, they wane out. Their effect wanes out. It probably wanes out right after you get dosed. The waning effect starts, but it's a gradual process. And at about 5 or 6 months after you're double vaccinated, you're still much more protected than somebody who's not vaccinated, but you're much, much less protected compared to after you just received your 2 doses. So Israel saw that and then made an emergency decision. Again, our models showed that if we give a booster shot, give a 3rd dose to people, we would be able to get to turn the curve and get out of this 4th wave without the need for strict measures. So that also came out of the modeling approaches, and Israel made a very bold decision, and it approved the booster shot before the FDA approved it. And actually, the FDA then learned a lot from the data of Israel and relied a lot on that data that came out in order to finally follow the decisions that were decided in Israel, and also approve the booster shot for quite a wide population in the US. And that booster shot has really turned things around here. And since September, we're seeing a decline, which is continuing. And my predictions now are that this decline will continue. And whether or not we'll have a fifth wave, I think we learned that it's very hard to predict so far in advance. We learned measures of modesty in this pandemic, so we don't know, but at least we know where to look at, and to look at effects of vaccines and waning effects and monitor that very closely. So that's kind of a summary of what went on here in Israel, the goods and the bads, where I think we really taught the world, which was a real privilege for a small country like Israel. Yeah, that's my summary.
Oh, that's a very interesting and wonderful summary. How would you summarize what were so that other countries can learn and reflect back on the steps that Israel took? What were the really important things that helped to control? What could Israel have done better or quicker? What were the goods and what were the bads? What can we learn from this?
Yeah. So again, I think the reaction to a pandemic is very different. The measures you need to take are very different when you have vaccines or when you don't have them. So we could talk about what happens when there's no vaccines, but we're not in that situation. So I think that's less relevant. I think I'll say just in one sentence that in retrospect, I think you have to act very quickly and disproportionately to the number of cases. Even for a small number of cases, when you don't have vaccines, you should take disproportionate, very strict measures to lower and control so that you won't reach the high state, because you'll reach it anyway when you don't have vaccines. But when you have vaccines, I think that the focus should be very heavily on vaccine administration. I think what we could do. So again, I mentioned the good, that the rollout here was very rapid and that it managed to block the wave. But I think we could have saved more lives if we were to do it even quicker, which we could have. And if you go into the populations that are typically of lower social economical status, that are more prone to hearing a lot of the fake news, and be under that influence, there's a lot you need to battle around that. You need to reach out and explain to people the benefits of being vaccinated, the very small risk that you take by getting a vaccine, how it was given to billions of people, and it's considered safe, and you really need to focus on your vaccination program, because it's such a powerful tool compared to all the other measures that one can take, which is why I think why I think that should be the focus. And right now, I think the biggest advice that probably could be given based on Israel's experience is to give a booster shot to everybody who's passed 6 months since their vaccine. This could really save lives and lower the pandemic.
Right. So that's regardless of age of the person. Right now,
yes,
in the United
regardless
States,
of age. Yes.
Yeah.
I think the booster should be expanded further.
Excellent. Interesting advice that you're giving. So we could expect a lifestyle where every 6 months people should plan on getting a booster if the government provide, the rules provide this.
So we don't. I'll say we don't know still how and if the third shot, the booster, wanes and decays in its effect. We don't know that yet. I think that's something to monitor. But so it is a possibility that it'll be every 6 months. I hope that it'll be maybe every year, maybe even longer. We don't know that still. But so I don't think we know, but I think right now we do know that getting the third booster is a good idea for all ages.
Yes. So I hear you also saying we have to recognize what we don't know.
Yes, absolutely.
And people want to know how come you're changing your mind, government, because
Yeah,
you're confusing
we're
us.
learning
but
new.
it's-
We're learning new things. We're learning new things as we go.
Yeah, excellent, excellent. Well, that is just a wonderful summary, Eran. It's really a pleasure to hear from you, your view, being right there with the government, your mathematical modeling of situations, their high accuracy, and some references you're making to the psychosocial status of people in terms of how we need to address various aspects of a population and reach out immediately to educate as much as possible. So this is just wonderful summary, and this is very helpful. I'm really delighted that you're here to share this with us. I would like to say a word to our participants. I'd like to suggest, when you have the time, to do an internet search on Professor Eran Segal, because you'll find that he does a lot of delightful interviews on topics of health and nutrition and things we're interested in. And we can learn further from him beyond the subject of today. And he also mentioned the interesting thing that he does tweet out findings of his research to help to educate everyone about the very interesting work that he does. I heard him saying, you have to have data, you have to have mathematical reasoning, and not just something you hear from the neighbor, or, you have to have data, which he provides and does wonderful work. And Eran, it was a pleasure to talk with you this morning. Thank you so much for being with us.
Thank you. Thank you very much. Very nice being there.
Take care.
Thank you.
Bye-bye.
Bye-bye.
Just as we have been talking about mutations in COVID, we are switching now to an Israeli discovery in mutations in another system, the cancer cell. And here to tell us about her research is Professor Yardena Samuels. Welcome, Professor Samuels.
Thank you for having me.
By way of introduction, so that we can know something about Yardena's background, she is an esteemed molecular cancer biologist, recipient of many coveted awards, and faculty member of the Department of Molecular Cell Biology at the Weizmann Institute of Science in Rehovot, Israel, where she is director of the Eckhart Institute for Cancer Diagnosis Research and director of the Weizmann Brazil Tumor Bank. She is the incumbent of the Knell Family Professorial Chair. A native-born Israeli, Yardena received her Bachelor of Science degree from Cambridge University in the UK in 1993, then served in the IDF as part of the Medical Corps labs. When she finished her tour of duty in the IDS, she worked for and received a Master's of Science degree in immunology from Hebrew University of Jerusalem, Hadassah Medical School in 1997, and completed a Ph.D. in molecular cancer biology at Imperial College, London in 2002. She was a postdoctoral fellow at Johns Hopkins University School of Medicine from 2003 to 2006, during which time she had two major findings in the field of cancer genetics. She remained in the US for a period of time, serving at the National Institute of Health as head of the molecular cancer genetics section before joining the faculty of the Weizmann in 2012. Professor Samuels is a member on several editorial boards and serves as the scientific editor for the journal Cancer Discovery. We're honored to have you with us today. Yardena, please start and tell us about your research.
Thank you very much, Ruth. That was a very beautiful and kind introduction. So I actually prepared a presentation that I will share with you now. Here we go. Okay, so my field of study, I'm a cancer geneticist, and we focus on melanoma, searching for new therapeutic targets. So this is just a few introductory slides talking about cancer and how it is the disease of the genome. And so on the left, we can see here normal cell division. I'm just going to use a pointer here. Okay. On the left, we see a normal cell division where when a cell gets damaged and doesn't get repaired, it will either just die by apoptosis or suicide. But on the right-hand side, we see a cancer cell division, which is basically uncontrolled cell growth. The cancer cell will acquire mutations. They will not die because of these mutations, and they will keep proliferating, ultimately leading to cancer. So this is just a reminder, in every cell we have a nucleus. These nuclei have chromosomes, which are basically our DNA, which will encode our proteins. And here we see an example of a mutated gene within the DNA, which will lead to tumor, potential tumor formation. And this is the first example showing that cancer is a genetic disease in a stage-wise manner. This is shown in colorectal cancer, where we can see here stage-wise morphological development of colorectal cancer, starting from these intestinal epithelial crypts to barren crypts to adenomas, ultimately carcinomas. And this was developed by Bert Vogelstein, Professor Bert Vogelstein at the Johns Hopkins, with whom I did my postdoctoral studies. And he showed for the first time that each one of these stages is linked to particular mutations in different genes. And this became a paradigm that cancers develop in a stage-wise manner on a molecular level as well. But in order to push this discovery further, we needed a few revolutions. The first was the Human Genome Project, where there were two initiatives to map the full human genome, and this became a template to compare the cancer genome and identify systematically cancer mutations. The second revolution is the sequencing revolution, which turned digital. And the moment it was digital, it was much more accurate, it was much more high throughput, and we could start looking for mutations systematically across the full genome. And I like showing this metaphor where you see this man looking for his keys. And when there's some light on the road, and if the light doesn't show the area of the keys, he won't find them. In the same way, in the past, when we used to sequence DNA, it was very particular regions, and then maybe we wouldn't find a mutation. But with the systematic analysis that we can do now, we basically light up the whole street, and now this man can find the keys and we can find mutations. Now we need tumor banks. What do I mean by that? We need large amounts of samples from different cancer types and establish these as a well-controlled bank. So we grow these either as cell lines in the lab. We take the tumors and we grow them in the lab, so we have a lot of material, or we put the samples, the tumors, in mice, so again, these can grow. And then we derive genomic DNA, and then we can sequence these. And so we derived such a tumor bank in collaboration with numerous clinicians around the world. Why are we doing this? Well, we want to make the medicine more precise. What do I mean by that? This is called personalized medicine. So if in the past, a particular category of patients was called by the pathologist a particular type of a cancer based on pathological attributes, now that we can sequence these tumors, we can subdivide them molecularly. And so we can now look at them in discrete manners, and these are actually different diseases, and we can treat them in a much more personal way and more successfully. So identification of genetic mutations will provide opportunities for accurate prognosis and new drug developments. And we decided to focus on melanoma. So this is a form of a skin cancer. So we can see here melanocytes, which are pigment-producing cells. I decided to focus on this cancer type back in 2006 because it's the most lethal form of skin cancer. Its incidence is all the time rising. The patient survival after late-stage diagnosis is only 6 months. Very little was known about the genetic basis of the disease. And here we see the sun exposure on a particular melanoma seen here. And like I said, the incidence just keeps rising. And so I decided to look at this cancer type and understand its landscape of mutations. And this is a slide from the Tumor Cancer Genome Atlas, which is a huge consortium that was set up to look at this landscape, and my lab was part of the consortium. It's a very busy slide, but the main point is that there are many mutations that we identified in melanoma, and it, excuse me, allowed us to subcategorize the patients to subgroups, and at least one of these has particular targeted therapy that could be given based on this identification. And here we see a flow of different types of treatment options for melanoma patients over the years. And you can see that about 12 years ago, there was a huge a revolution in the number of types of treatment options that we could give. And we see here some targeted therapies, meaning that they're specific for particular mutations. But we also see immunotherapy and the combination of the two. So the problem is, of course, resistance. So we see here a patient who has metastatic melanoma who was given this targeted therapy, so a BRAF inhibitor or a combination. And you see how impressive the results are. It's really within weeks you can start seeing response. Unfortunately, about 6 months later, there's progression again, an extremely aggressive progression. And so these are resistant clones. So on one hand, we're understanding the disease much better. On the other hand, there's much more work to be done. Unfortunately, immunotherapy emerged and was given a Nobel Prize in 2018. So meaning checkpoint inhibitors. So these are particular antibodies, and we'll look at them more closely. But the whole field really underwent a revolution thanks to this. So we see here a melanoma cell interacting with a T cell, which is a cell from the immune system. And we see here Keytruda, which is one of these checkpoint inhibitors who received the Nobel Prize, which basically inhibits an interaction between the melanoma cell and the T cell, and allows the T cell to be extremely active against the melanoma cell. But we need to remember that in order for this to happen, we also need the T cell via the T cell receptor to recognize the melanoma cell through the peptides that the melanoma cell is presenting on what are called HLA molecules. We see it right here. So if this is like a lock and key interaction. And if this interaction occurs, there's a chance that the T cell will be able to kill the melanoma cell. And this is true also for other cells in the body. So if there's a cell that's invaded by a virus, those viral proteins or peptides will be presented on the HLA molecule, and then T cells will be able to kill that target cell. So my lab is extremely interested in identifying these particular presented peptides. So how are they brought up to the surface? Well, here we see the DNA, which is made encoding a protein, which is then degraded into peptides, and these peptides are shuttled into what is called an endoplasmic reticulum, where it will bind this HLA molecule, which gets shuttled up to the membrane together with a peptide, which is presented to the T cell. So again, if this DNA has a mutation in it, like in a cancer cell, there's a chance that there will be a mutated protein expressed. There's a chance that there will be a mutated peptide, which will be presented on the surface of the cell. So we call this a neopeptide. And because it's different from other peptides in the body, because this is derived from a mutation in the cancer cell, it labels that cell as a cancerous cell, and we can use this for treatment. So here we see a melanoma cell in green, and T cells from the same patient interacting with a melanoma cell and killing the melanoma cell very efficiently, ultimately killing all these melanoma cells on the plate. This is exactly what we'd like to see in a patient. And therefore, we need to understand this interaction much better. So this is what these T cells see. So we see here an example of a melanoma cell presenting these HLA molecules with a peptide within them. You see the green, the pink, and the blue. These are peptides, different peptides. The T cells come over and kill the target cell, the melanoma cell. So we want to identify these neopeptides. How do we do it? So we collaborate with many clinicians around the world and get these tumors from melanoma patients. We identify the mutations that these tumor cells have. So here we extract the DNA. We now know what mutations there are. And we now extract these actual HLA molecules and the neopeptides bound to it. We elute those peptides and we run it on a machine called a mass spectrometer. Once we run it on this mass spectrometer, we can identify which peptides we want to target. How do we target them? There are several ways. One could just vaccinate with the actual peptides. This is a cancer vaccine. Or you can express them as an mRNA molecule, just like we get the COVID vaccine as an mRNA, we could express these neopeptides as mRNAs. Or we could identify the actual T cells that interact with these neopeptides and then use them and transfer them back into the patient. And we can even engineer T cells to express the T cell receptor that recognizes the neopeptide and put these back into the patient. And this is not fiction, this is actually being done in the clinic. So this is an example of a successful treatment by Dr. Steven Rosenberg at the NCI, where he identified T cells that recognize a neopeptide from a KRAS mutation, and the treatment was extremely effective. The metastases regressed. So this is just some switching gears here, and I'm showing you that we can identify also antigens derived from pathological states such as bacteria. So here we see a tumor that has been invaded by bacteria. And we've seen this before, it's been published, bacteria can reach tumors, but no one has ever shown whether these bacteria can enter the tumor cells and be presented in HLA molecules. And we did show that. So we see here melanoma cells that are fluorescently marked, and these rod-like structures, these are bacteria that we can see actually entered the melanoma cells. And when we searched, we actually identified peptides that are presented by these HLA molecules. This was published this year in Nature. And here, this is just an Andy Warhol, this was inspired by Andy Warhol's picture of Marilyn Monroe, where we showed different melanoma cells invaded by bacteria. So we can find different types of peptides that are being presented by HLA molecules. And we recently identified one that is recurrent. It's presented by many different patients, melanoma patients. And so this is a neoantigen that can be used in the clinic for thousands of patients every year. And we're looking into doing a clinical trial with this particular discovery that we made. And again, we published it this year. So we know quite a bit about cancer immunogenomics, but this is still the tip of the iceberg. We're working very hard to identify more of these neopeptides and their T cell receptors. But this is definitely an immunogenomic era. Clearly, there's a lot more to be done, and it's very successful and very, very exciting. So we have two models to improve melanoma therapy. One is to target the host by immunotherapy. The other one is by targeting the tumor by targeted therapy. It's probably the combination of the two that will work the best. So if we look at what we've had for many years, chemotherapy, this is the kind of response and patient survival expected. When we start targeting genomically altered genes, within the tumor, with targeted therapy, this is the kind of survival. With immune checkpoint inhibitors, immunotherapy, we increase the survival to longer. We get an overall longer survival and durable response. We expect that a combination of immunotherapy and targeted therapy will improve the median overall survival as well as a durable response. The main thing is now a lot of clinical studies are being done combining immunotherapy and targeted therapy. The bottleneck, of course, is analyzing all this data because we need to look at the biomarkers, we need to look at the response, but we're getting there. And numbers here are key because, the more of these patients that we treat, the better we'll see these pixels, and we'll be able to tell if this child is smiling or frowning. And so we're really looking to expand as many clinical trials as possible to find out which are the best combinations to treat patients. I'm switching gears slightly to touch upon viral infection, taking into account the pandemic. So here we see a virus entering a cell and expressing its genes and its proteins. And these proteins also get degraded and get presented on the HLA molecules, eliciting a T cell response. And so this is what you would expect to see, for example, if SARS-CoV-2 COVID enters our cells. So these are the different peptides that may be presented on the HLA molecules of the cell. And when the pandemic began, almost 2 years ago, 2 of my doctoral students came to me and said, let's use our pipeline to try and help the COVID situation and identify the peptides that are being presented. And I agreed, and we started looking for SARS-CoV-2 presented peptides. This is the pipeline that we used, and we found quite a few, many more than are being targeted today with the vaccines like the spike. So these are the ones that we identified, and we also found that some of these are immunoreactive, and we took blood from patients who had severe disease. And basically what we see here is the reactivity of these T cells against some of the neopeptides, the peptides that we identified. And these are the Ph.D. students that did the work at the Shelley, and this is the paper that we published. The conception was in early March 2019. So I'm showing you this because I'm not the only one. There are many scientists that switched gears the moment the pandemic began in order to use their pipelines in order to help the situation and try and find out more in order to provide more data for vaccination. And in this case, we have different vaccination strategies. As you know, the one that a lot of us are receiving is the RNA vaccine, which is expressing the spike protein, which of course limits the entry of the virus into the cells. But based on our data, we think that possibly it may be worth also expressing other genes from the virus, because these are also presented as peptides on infected cells. So I'm coming to the end of my talk here and my presentation, where I'm really showing here the different types of peptides that we've been able to identify over the years, whether it's neoantigens, bacterial peptides, viral peptides. There's a lot of black matter, dark matter that is still to be found out, and we're working hard to identify that. And if we look at a melanoma cell, you see these various different flavors of peptides are being presented upon the surface, some of which are immunoreactive. And so this is really giving a whole landscape of different peptides, and we really need to understand what they're doing there and how we can take advantage of them for treatment. So I'll end here with a quote by Dr. Keith Flaherty from MGH, who's a very well-known melanoma oncologist, who said, after more years of doom and gloom than I'd care to count, we've had this amazing trajectory that doesn't seem done yet. Our confidence keeps rising as our patients keep surviving. And it's true, we're really living a revolution in the field of therapy, cancer therapy. This immunotherapy is really making a difference and helping a lot of patients around the world. This is my great lab who are really exceptional, worked extremely hard during the pandemic and published. And they are the talent behind all of what I showed you today, and many, many collaborators, and of course, funding sources. Thank you very, very much, and I'll be happy to take questions.
Yardena, that's just wonderfully amazing, just wonderfully amazing. It's great to understand, for us to understand more about cancer as a genetic disease. Admire all the collaboration, the jumping on the using your techniques to help with the COVID and the immunotherapy involved in the progress in the digital age. I'd love to have you go on and on.
Thank you very much.
What advice would you have to people in a general way about, let's say, melanoma cancer? Their mutations occur. We hear about staying out of the sun with melanoma. Are there advice in this regard that you could give to individuals?
Yeah, I think that's the best advice. As I'm not a clinician, but you know, avoiding the disease is always better than treating it. And so avoiding the sun, you know, at particular hours for sure, and dressing appropriately for the sun, especially if you're fair like me. It's a disaster. So a hat and long sleeves probably, but you know, and then going for regular checkups. That's important because early diagnosis is key here. So you know, if you can find it early, then that's the best case scenario, and just immediately deal with it rather than postpone any you know, any clinical steps. That would be the advice that I would give. And of course, if God forbid there's a later stage disease, then you know, go and get as many opinions as possible and treat as quickly as possible.
That's wonderful, wonderful. That's very, very helpful. We noticed a Hadassah connection. Do you have any further comment about you and Hadassah connection?
Yeah, so I did my master's at Hadassah, and I also have a personal connection on the level of my mother, who established Hadassah International in the 1980s. And so I remember it as a child. Very, very impressive, where she worked extremely hard to establish lots of connections between Hadassah and various collaborators around the world, including the Pasteur Institute, Institut Curie, and bringing donors to Hadassah in Israel. And very proud of her.
Oh, well, thank you so much, Yardena, for sharing that with us. This has been a wonderful joy. And thank you so much for your time, your gracious manner. This has been very, very interesting.
Thank you.
I want to take, yes. I want to take this time at the end of our production here to thank, give our thanks to many people. First of all, I want to thank the Joan and Stanford Alexander Israeli Programming Fund for partially funding this program. I want to thank Stanley Horn and Dr. Sharon Rome for working very hard, making calls to line up people to talk with us. I want to thank Laura Kaplan, Beth Israel staff member, for the publicity. I want to thank David Scott, Director of Lifelong Learning and Engagement. Nothing would work here without David. It's just very, very much appreciated, David. And Senior Rabbi David Lyon for his guidance and input into the Israeli Advocacy Committee. And thank everybody who joined us today. This has been a wonderful experience. Thank you all.